So we went through our blood cells. Then with white blood cells, that catapulted us to the immune system. Well, now we're going to bounce back to blood cells again for just a few minutes before we get into the bigger aspects of our circulatory system, especially the heart and how the heart functions. So as we look at red blood cells, we're very keenly aware of blood typing, especially in clinical settings, because if you don't have the right blood type and someone gets a transfusion, that could be catastrophic. And while there are many different blood groups, the one we are most familiar with is the ABO blood type group. Now, if you don't know your blood type, I would suggest you go out and you find out your blood type either at the doctor or when you donate blood or plasma, they will determine your type, because that's going to be important information that you need for the rest of your life. And when I first learned about blood types, and you may have been under this misconception as well, that our ABO blood groups mean you have the A antigen on the surface of red blood cells, you have the B antigen, or you don't have an antigen. If you've got O-type blood, you don't have that marker on your red blood cells. And that's only partly true. Because when we look at O and A and B, these different blood groups are all basically facilitated and mediated based on a foundational receptor on the surface of your red blood cell that's called antigen H. Antigen H is a glycoprotein. It's got protein, it's got some carbohydrate on it sticking out on the extracellular surface. And if you have an unmodified antigen H, you have O-type blood. If that's all you have on the surface of your red blood cell is unmodified antigen H, you're O-type blood. However, if you have antigen H that has an extra N-acetylgalactosamine, one extra sugar group placed on it, then you have A-type blood. Or conversely, if you have the H antigen that's got an alpha galactose added to it, you've got B-type blood. So you see, in fact, it's not the absence of an antigen, it's the absence of the extra sugar that makes either the A or the B antigen. Now again, remember back to our blood group genetics. You have two alleles. You have an allele from mom and an allele from dad. So if you have O-type blood, that's your phenotype, what is expressed, and you only have the H antigen, that means you got an O allele from dad and an O allele from mom. It's the only way you can have O-type blood. Conversely, if you look at the surface of your red blood cells, if your phenotype is AB blood, that means one parent gave you the A allele, so some of your receptors, some of your H antigen is going to have that extra N-acetylgalactosamine, whereas the B allele you got from the other parent, and so some other of your H antigens are going to have the alpha galactose. You're not going to have the two extra sugars on one H antigen, but you will have both the A substitution and the B substitution. For A-type blood and B-type blood, depending on your genotype, you could be AA or AO. That's going to determine how many of the A antigens are going to be present. If you have AO type blood, half are going to be A's and half are going to be O's. But O is not antigenic. You're not going to have any antibodies in the human body that are going to recognize just the H antigen. But it will recognize that extra sugar that converts it into either an A or a B. And it's the antibodies then that cause the problems. Because we already know with antibodies, you can handcuff pathogens together. You can create these huge rafts of pathogens. Well, if you have antibodies against your blood groups, well, you're going to start tying your red blood cells together. And don't forget, when we talked about isotypes of antibodies, it was the IgM type that's primarily going to be responsible for this. Remember the IgMs, the pentamer? 10 binding sites. So you're going to clump the blood together excessively if you have a mismatch. And if this happens in the circulatory system of a patient, it's going to be disastrous. Heart attacks, strokes, embolisms. Yeah, it's not going to be good. So in our bodies, we have either or anti-A antibodies or anti-B antibodies or both. And it's going to be the opposite of the antigens we have on our red blood cells. Do you know what your blood type is? A positive? O, I'm A positive, I think positive or negative. So for our A-type blood, we only have antibodies that recognize B blood. For you, O-type, you have antibodies that recognize A and other antibodies that recognize B, because you have neither. Whichever antigen you have present, that's the antibody that you won't have in your plasma.
Is that why that's considered the universal donor?
Yeah, we're getting there, but I got bad news for you, though. That's coming. So that's how the antibodies work. Now, here's something that I want you to think about. Whenever, in the last chapter, before we encountered the chickenpox virus, or before we got our chickenpox vaccine, did any of our bodies make antibodies against chickenpox? No. We had to get vaccinated or exposed to it to have a primary immune response to start making antibodies. Well, we have A-type blood. You have O-type blood. You've got AB antibodies. We've got B antibodies. Have you ever had a blood transfusion? Have you ever gotten the wrong type blood? Have you ever had a blood transfusion so that you've been exposed to A or B type blood? How do we have these antibodies? To stuff that we've never been exposed to. No, it's not inherited. It's not inherited. In fact, it's believed that it could be some environmental agent that we encounter that leads us to develop antibodies that cross-react, that are similar enough to our A or B blood groups that cause us to have those antibodies. But to me, it seems like if it's that random, why aren't we making antibodies against our own antigen types? So I haven't wrapped my head around that. That's called the light in the dark theory, and I think we've got that coming up in a slide or two. But this illustration gets at the difference between your antigens that you see across the top versus the antibodies that are present in your plasma. Again, the antibodies and antigens will just be the opposite of one another. Yep, there's light in the dark. So viruses, they infect cells and they turn the cell into a viral factory. And so some of these viral particles are going to have receptors on them that they bring with them from the host cell. And so it's possible then that some of our membranes have receptors enough that they cause an immune reaction that can recognize some of these antigens. But again, I don't see how we're here. We don't have autoimmune reactions to our own red blood cells more often. And so, I mean, this was proposed back in 98, which is crazy because that was almost 20 years ago. I read something the other day, we're closer to 2047 than we are to 1989. Something like that. Crazy. So, the big answer to this, we don't really know yet how these blood antibodies really do develop. But we know that they can cause clumping, which is called agglutination or coagulation of the blood. Looks like clotting, but this is this mass of red blood cells tied together with antibodies. And so clinically, there's something that happens called a major crossmatch. Before you give someone a transfusion, especially in the surgical suite, what you can do is you can take some of the donor's blood, principally the donor cells, and you take the recipient's serum, because that's where the antibodies are by and large going to be that's going to cause the problem, and you mix them together. And when you mix them together, see if they clump up. If they clump up, don't give the patient that blood, because there wasn't a match. Now notice, you've got to do this in the presence of EDTA or citric acid, because if you don't use EDTA and citric acid, when you start stirring the blood up, you could get a blood clot started, which is going to look like this agglutination coagulation, and you could be fooled. You could think that, oh, it's not a match, when in fact, it really would be. Here in these 3 test tubes, we see sort of different levels of coagulation. The one on the right, do you see these clumps, these dark red clumps in the bottom of the tube? This would not be a match. So the red blood cells are clumping together, and they're basically precipitating out of solution, leaving the serum present. So if you happen to see something like that, you would know somebody has antibodies against the red blood cell markers. Now we even utilize the coagulation reaction for even typing your blood. I think they used to do this in genetics class. I don't know if they still do. But you can take ready-made antibodies, mix them with your own blood from a finger stick, and put it on a card like you see here. In this circle, this was a drop of blood that was mixed with anti-A antibodies. This was anti-B antibodies, and we haven't talked about Rh factor yet, but that's going to be in that 3rd spot. So if you got agglutination in the A spot, nothing in the B, and agglutination in the Rh factor spot, what type blood does this person have? A positive, because it's only going to agglutinate if the antigen is present on the red blood cells. The B antibodies didn't cause clumping, so there's no B antigen. So in this top slide, no agglutination for A, no agglutination for B. We do have agglutination for Rh factor, so this person has neither A or B, so they have to have O-type blood.
Is there such a thing as A negative blood?
Yes.
And
Negative,
positive or negative?
negative is more rare in the human population, but again, it's going to be using antibodies to identify that particular factor. But positive is much more common than negative, as we'll see right now as we talk about. That was a great segue as we get into Rh factor, because this is a separate blood group. There are a number of different alleles for Rh factors. They have dominant C's, dominant D, E's, but really they look at the RhD predominantly as the allele that sort of determines if you are Rh positive or Rh negative. You either have it or you don't. And Rh positive has a number of these alleles that can result in the positive type, the presence of Rh factor. And so that's why, by and large, in our population, it's hugely dominant. 2, the negative, which has to be homozygous negative, and there's only one allele that can really facilitate that phenotype. So that's why our positives drastically outweigh our Rh negatives in the population. Now, here's another reason this whole light in the dark theory, I don't understand it. And I understand that ABC blood type, ABO blood type, because it's that extra sugar, there's a whole different set with the Rh factor. But you never have ready-made antibodies against Rh factor. If you are Rh negative, you don't have Rh antibodies. You know, like if you're O negative, you got A and B antibodies. Rh factor, Rh negative, you don't have antibodies to Rh factor. So it seems like it would be the same for that if that hypothesis really worked. And so by and large, the only time that you are going to see antibodies against Rh factor is if you have been previously exposed to Rh positive blood. Now, if you have a positive blood type, you don't have antibodies against Rh factor because that's your own blood type. And really the only clinically significant time that this may happen is when you have a female patient that is Rh negative and she has the potential of having a baby that is Rh positive. Now, if baby daddy is Rh negative, you got nothing to worry about. But if baby daddy is Rh positive, that means there's at least a 50-50 shot of the baby being Rh positive as well. And during the birth process, an Rh positive baby's blood is going to mix with Rh negative mama's blood. If you've not experienced a birth or seen a birth, it is a bloody mess. Just saying. It will ruin your day if you're not prepared for it. And so for that first baby, the baby's gone, the cord's cut. Baby's fine. But now mom is exposed to Rh positive blood. Mom's going to have a primary immune response. She's going to make antibodies against the Rh factor. So if there is a subsequent pregnancy, a second Rh positive baby years down the road, those IgG antibodies that she's making against Rh factor, we know they can cross the placenta. Those antibodies could attack the red blood cells of the developing fetus. And it could lead to our condition here called erythroblastosis fetalis, or hemolytic disease of newborn. And it can be a fatal condition. So how is that prevented? Well, when you have an Rh negative mom and she's pregnant, just before she goes into the delivery room, if not a day or two before, she gets an injection of a substance called Rogam. Rogam is ready-made antibodies against Rh factor, so that if any of baby's blood is mixed, again, just a statistical chance, there's much more likely a chance that that artificial antibody is going to bind to the Rh factor than mom's own B cells, and so she will never have a primary immune response. And mom's got to do that with every pregnancy if she still wants to continue to have babies. If mom's going to have that baby number one and that's going to be it, and she's going to have her tubes tied or a hysterectomy, well, you don't need Rogam. But if she's going to have the potential of having a baby down the road, Rogam especially is going to be important so that her antibodies don't attack the next baby that she incubates. So here are our population breakdowns, and you see how positive is vastly. I mean, look at positive. We've almost got 90% of the population. I'm not doing my math so fast in my head, but I think this over 90% of the population is going to be Rh positive. And you were O positive? O positive? Yeah, that's the most common type of blood that we have. And here we are only in second place. So we are so common with our blood types, it's crazy. But here's the thing. You were mentioning universal donors and universal recipients. Well, a donor is going to have those cells that can be given to anyone. And you're close. You're almost universal donor. The only problem is you got a positive, and it's the O negatives that are the universal donors. But see, as O, you can give blood to any positive blood type. You can give blood to A, B, and O, as long as it's positive. So you're semi-universal. But you can only have blood from O positive or O negative people, because everybody else is going to have the A or the B antigen your antibodies aren't going to like. So I guess in our selfish society, it would be much better to be the universal recipient, which are going to be our AB positive individuals. Because if you're AB positive, you have no antibodies, nor do you have the capacity to produce any antibodies against any antigen. So you can take blood cells from anybody. But the problem, as you get so general about that, it's not so much about the antigens on your red blood cells. But let's say, for instance, you're an AB positive person and you're in an emergency situation and you need blood, and they say, look, the only blood we have out here is O positive. Well, they're AB positive, give them O. Well, you can give them O, but you can't give the whole blood. Because if you give a unit or two of whole AB positive blood, of O positive blood to an AB positive person, what else are they getting in that whole blood? The AB antibodies that are present in the plasma. So when we look at our plasma, we're largely going to see that the ability to accept the plasma is going to be the opposite of what we see with our red blood cells. And in fact, it's because of those antibodies in the plasma that you can't accept whole blood from just anyone. What they will have to do in that situation is they will have to centrifuge the blood so that they spin the cells down, get rid of the plasma with all those antibodies against A and B groups, resuspend them in our isotonic saline, and then give that to you. That would be the only way that it would really be safe. And so with our plasma, we see whereas O, and we don't have our Rh factors here, right? Because we're not going to see antibodies against Rh factor. We're just not. Unless you're a Rh negative mom and you had the baby, blah, blah, blah. So here, if you are an O person, you're going to be able to receive blood from anybody. Because you've already got the antibodies, and not blood, but plasma. Anything you get from anybody else is not going to have any antibodies you don't already have. But if you're AB, that plasma, you have no antibodies, so you can only get plasma from AB. So when you look at transfusions, it's a little bit misleading in, yeah, you need the cells, but you can't have all the blood depending on that type. So those AB positive universal recipients, yeah, they are until they start getting a lot of plasma, and then it's a different issue. They have to be, and really they're going to try to match your blood type and your plasma type regardless. They're just not going to willy-nilly give you anything on the shelf. Now, this is so really cool that even though it was 10 years ago, probably something you've not even heard of, but I mean, blood supply is usually short. I mean, when you look at blood centers, they're always saying, we need more blood, especially when disasters are looming. And so since blood is in short supply, wouldn't it be great if you could take any blood type and convert it into O-type blood so anyone could have those cells? And guess what? You can absolutely do that. Because what did we say the difference was between O and A, or between O and B? One sugar. One sugar, one extra sugar. Well, they're antibodies, enzymes that can remove carbohydrates. And so there have been glycosidases that have been identified that can remove those one sugars, leaving the H antigen and converting the type of blood to O. And that was back in 2007. I think this was also in a Nature magazine. Why haven't we heard of this more? Why aren't they doing this routinely in blood centers? Somebody discovered it, they patented the enzyme, and you can do it, but you got to pay for it. It's all about show me the money, right? Isn't that what it's all about? So again, it's like, duh, why? It's so simple. Why hadn't this been done a long time ago? Well, patents. Patents, patents, patents. So here's a cool little genetics problem to get at that phenotype-genotype issue. So let's do this sort of genetics problem. We've got 4 children, one set of parents. Each child has a different type of blood. Is that possible for one set of parents, or do we need to book Jerry Springer? And so let's kind of work backwards from what we know about the phenotype of the children. Now, we know the phenotype of our AB child is the same as the phenotype, right? So AB, that's genotype and phenotype. We also know that O-type blood is also the genotype, right? Because the only way you can have O-type blood is if you have homozygous O. So A and B, you got an A from one parent, a B from one parent. You've got homozygous O in this child, so you had to get an O from each parent. So now we see the genotypes of our parents. Can we determine the genotypes of the other two siblings? This child has A-type blood. Well, they had to get A from this parent. So which allele did they get from the other parent? Had to be O, because if they got the B, they'd have AB blood too. So we see the genotypes, and it is absolutely possible for one set of parents to have 4 children, each with a different blood type. Now, the only way this wouldn't work is if this parent was homozygous A. If this parent was homozygous A, and we got an O-type child out here, yeah, somebody has got some explaining to do. So that's how the genotypes, phenotypes, and how in your question can you determine the baby's type. It's a lot less invasive and easier if you just look back at your parents and grandparents, and you can probably get a really good idea of what your genotype is just from the inheritance of our ABO blood groups.